Hello and welcome to Neuro Navigator. Today's topic is visual processing. Same as with the action potential, this topic is broken down into multiple parts. In the previous videos, we've talked about eyeball anatomy and photoreceptor transduction, and these next videos will talk about where that information goes. Let's get started. In the 1950s, Stephen Kluffler recorded signals from ganglion cells. His research has allowed us to understand that there are different types of cells in the visual pathway that respond to light and darkness in their receptive fields differently. So we'll be talking about on and off center cells. Last video talked about photoreceptors hyperpolarizing and not releasing any glutamate when there is light in the on center of the receptive field. So how does this information get communicated down to higher brain areas? Well, not so simply. The photoreceptors feed information to an on-center and an off-center bipolar cell. The on-center bipolar cell has mGluR6 receptors, which is a metabotropic receptor that closes with cGMP-gated sodium channels when glutamate binds. The off-center bipolar cell has AMPA and kinase channels, which are ionotropic glutamatergic receptors, which allow sodium and potassium through when glutamate binds. In our example, with the hyperpolarizing photoreceptor, neither of these cells will receive glutamate. The off-center bipolar cell won't send an action potential, but let's observe what is happening in the on-center bipolar cell. Because glutamate isn't binding, those cGMP channels are going to stay open. The cell will depolarize and send glutamate to the next cell in line, which are called on and off-center ganglion cells. The so-called on-center ganglion cells respond with increased firing rates when light is in the center of their receptive field. Conversely, the off-center ganglion cells respond best when there is a decrease of light in the center. Both of these cells have AMPA, NMDA, and kinase receptors, and all of those are ionotropic glutamatergic receptors that allow sodium and potassium in whenever glutamate binds to them. The off-center ganglion cell receives no glutamate, but the on-center cell does. The glutamate binds to the AMPA, NMDA, and kinase receptors. We can apply the same logic if we say that this photoreceptor is in the dark. In that case, the opsin and the retinol do not change, the cGMP is not hydrolyzed, and the cell will depolarize, releasing glutamate into the synaptic cleft. When glutamate is released, it causes the on-center bipolar cell to hyperpolarize as the cGMP channels are closed, while the off-center cell will depolarize as glutamate attaches to its AMPA and kinase receptors, sending glutamate and a depolarizing influence to the off-center ganglion cell, causing that cell to depolarize. Where does it go next? Well, you can either Google it right now, or you can follow along to find out.